Hello students, welcome once again. Now I am here in front of you with the summary of differences. As we have discussed about the prokaryotes and eukaryote cells in our last video, you have already gained the knowledge about the prokaryotes, especially bacteria cells. Now we will discuss further about the differences. As you are watching in the table, the prokaryote cells and eukaryote cells differences. At first we will discuss the small size and large size. It means we differentiate on the basis of their size. Prokaryotes are smaller size than the eukaryotes. Prokaryote cells always unicellular while eukaryote cells often multicellular. Prokaryotes have no nucleus or any membrane bound organelles, while eukaryotes always have nucleus and other membranes bound organelles. DNA is circular, found in the prokaryotes without histone protein, while DNA is linear and associated with the proteins in the form of chromatin in eukaryotes. Ribosomes are found only 70S types, while in eukaryotes ribosomes are found 80S larger. There is no cytoskeleton present in the prokaryotes, and while they always has the cytoskeleton in eukaryotes. Cell division is by only binary fission in prokaryotes, while the cell division is by mitosis or meiosis in developed eukaryote cells. The reproduction is always unisexual or asexual. Asexuals and reproduction is asexual or sexual, or both types are found in the eukaryotes. So okay students, is it clear? This is the plant cell. You can draw this diagram in your fair notebook. Each and everything cell organelles is mentioned neat and clean, so you can zoom in or zoom out and make with label diagrams in your notebook, fair notebook. And we will discuss now differences between the animal versus plant cell. You can watch here by the ray diagrams that there are some major differences between plant cell and animal cells with the diagrams. Plasma membrane, endoplasmic reticulum, mitochondria, nucleus, Golgi apparatus are found in both of cells, plant cell and animal cells. But in plant cell, only cell wall, chloroplast, large vacuoles are found, and in animals, centrioles and centrosomes and some small vacuoles, cilia or flagella are found. So these are the main differences between the animals and the plant cells. You can observe here very well. Now we will differentiate also with table. So you can see here all the differences which I have described you just. Now we comes on the animal cells. There are a lot of differences animal cells that carry out specialized functions. Therefore not every animal cell has all types of organelles, but in general animal cells do contain most of the following organelles such as mitochondria, Golgi apparatus, endoplasmic reticulum, vacuoles, peroxisomes, microtubules, ribosomes and nucleus. So these are the cell organelles which we have drawn in this figure. So please draw it neat and clean in your fair notebook, and after drawing we will discuss and write in your notebook about all cell organelles. At first, nucleus. As you know, nucleus was discovered by Robert Brown, but it is a behind story about the Franz barrier in 1804, and more details about the botanist Robert Brown. The nucleus contains DNA in the form of chromatin can be further composed of to form chromosomes. Nucleus is surrounded by double envelope that contain process to allow certain material to pass in and out. Nuclei also contain a region called the nucleus. So you can see this diagram of nucleus neat and clean. There are so many pores which allow to nucleoplasm and cytoplasm to come in and out. It is double membrane cell organelles, outer membrane and inner membranes, and inner side chromatin, chromosomes, nucleolus, nucleoplasm are found. So please draw it neat and clean. Then we will discuss ribosomes. Ribosomes founder George Emil Pallade. Ribosomes are organelles found in both prokaryotes and eukaryote cells. They are like mini machines that synthesize all the protein in the cell. In any single animal cell, there can be as many as 10 million ribosomes. The ribosomes from the manufacturing department of the cell. So ribosomes are the protein factory where protein is synthesized. And this is ribosome structure, which is made by the two subunits, a small subunit and large subunit. At about ribosome's diameter is 10 nanometer, and ribosome is attached with mRNA to synthesize protein. In prokaryotes and eukaryotes, you can also differentiate it on the basis of the ribosomes. 70S types ribosomes are found in prokaryotes, while 18S types ribosomes are found in the eukaryotes. And we can also see here about subunits of the ribosomes. In 18S, there is no mention subunit, so I am asking you 40S plus 60S is equal to 80S. And about 70S also, I want to tell you that 30S plus 50S is equal to 70S. And accept it, 40S, 60S also found in eukaryotes. Now we comes on the endoplasmic reticulum. The endoplasmic reticulum was discovered by Kath Porter in 1953. Endoplasmic reticulum has two types: RER, rough endoplasmic reticulum, and smooth endoplasmic reticulum, SER. Rough endoplasmic reticulum found ribosomes are attached to RER, and which synthesize protein. Smooth endoplasmic reticulum synthesize lipid and steroids, metabolize carbohydrate and steroids, regulate calcium concentration, drug detoxification, and attachment of receptors on cell membrane protein. This is the structures of endoplasmic reticulum. So students, is it clear? If you have any doubts, you can call or SMS me. Now we comes on the Golgi complex. Golgi body is combination of cisternae, lumen, vesicles. It has two phases: trans phase, lower side, and upper side, cis phase. Cis phase is inwards, and trans phase is outwards. Golgi body modifies protein and lipid, process material to be removed from the cell, make and secretes mucus, and for package products into vesicles for transport. So its main function to transport the material from one place to another to the outside of the cell membranes. So okay students, in further videos we will discuss many other cell organelles. So now it is clear that cell is the basic chapter for biology, and we have discussed all about it. As you have clear about the cell at first, then facts, overviews of animal cells, then about the largest and smallest cells, common cells types structure, prokaryotes and eukaryote cell structure, classification of cells, bacteria cell, summary of the differences between prokaryotes and eukaryotes, about plant cells versus animal cells, their differences in the form of table, then animal cells label diagrams, and then cell organelles, nucleus, ribosomes, endoplasmic reticulum, and Golgi complex. So okay students, in next video we will discuss more cell organelles. Stay at home, be safe, goodbye, bye bye, Jai Hind, Jai Bharat.